Now it's my pleasure to welcome and introduce our expert in blood cancer immunotherapy, Dr. Marcel Brink. Dr. Brink is head of the Division of Hematologic Malignancies and the Allen Houghton Chair in Immunology at Memorial Sloan Kettering Cancer Center in New York City, where he studies new strategies to improve immune therapies for cancer patients. While Dr. Brink is speaking, you can put your questions for him in the Q&A box. Brian Brewer from the Cancer Research Institute will be sharing them with him as we go along. Dr. Brink, Brian, let's learn about blood cancer immunotherapy.
Thank you very much, Tamron, and thanks very much to CRI. It's a great pleasure, a great honor to be part of this meeting and to tell you something about what I would call some of the most exciting breakthroughs in all of cancer. And why do I use that bold statement? Well, for many years, as probably most of you will know, the only therapies that we had were surgery, radiation, and chemotherapy. And now, since about 15 years or so, we have seen a breakthrough that is based upon trying to figure out how your own defense systems, your immune system, can actually also be used, can be armed as a way to fight cancer. That was a quest that many people had been working on for, well, you can easily say 150 years or so. And for many decades, we didn't see much coming out of all of these efforts. Then in the 1970s, for the first, we saw some glimpse that there was maybe something like immunotherapy of cancer. And we saw that within the context of allogeneic bone marrow transplantation. What is that? That means that basically to overcome all of the dangers, the toxicity of high dose chemotherapy, you rescue somebody's bone marrow with the bone marrow from somebody else. And people did that only to be able to give high dose chemo. But what they found out is that there was more to it. Namely, that within the bone marrow graft, that there were certain T cells, lymphocytes, that could recognize the cancer cells of a patient. That was called graft versus tumor, or GVL, graft versus leukemia. That was the first hint that we had that T cells could really kill a cancer. Fast forward to about, 15 years later or so, 20 years later, when for the first time people started to understand that T cells are being regulated in many different ways, that there are all kinds of pathways that block them and that can activate them. And people figured out if you block those blocking pathways, block the blockers, that is one of the tricks actually that cancers use, activating those blockers. Well, if you unblock them, then you unleash T cells that were trying to kill a cancer to now actually do it. Meanwhile, people had been also working on antibody therapies. So you have T cells and you have B cells. T cells work directly by recognizing cells with viruses, fungi, cancer in it and kill it. And you have B cells that can make antibodies. And that B cell repertoire can also be used to recognize and to kill cancer cells. And I'm going to show you some of those kind of examples. Finally, what we will first focus on is a cell therapy where we take the same T cells again and we engineer them to get better at recognizing a cancer and to kill it. So let's start with these CAR cells, chimeric antigen receptor T cell. How does that work? We start with isolating from a patient white blood cells. We literally just filter it out of your blood. At that point, in a culture flask, we engineer all of the T cells that are part of that white blood cell pool to contain certain molecules so that they can better recognize a cancer. I will show you what kind of molecules those actually are. Then you grow up all of these cells further and you give them back. That is your CAR cell therapy. That is being done now all over the world. So what is a CAR? So I said already, chimeric antigen receptor. What it does is it creates a T cell that can recognize an antigen as if it is a B cell. So, how does that work? You put a molecule into these T cells where the business end, the end that can recognize an antigen on the outset, is actually taken from an antibody. The inside, however, is still exactly the same as the T cell receptor. That is the molecule that a T cell normally uses to get activated. So a CAR cell recognizes an antigen with an antibody um, as if it is a B cell, but it gets activated in its own way as a T cell. Another way to make T cells that are very active is by putting in a T cell receptor, TCR, that can recognize a specific antigen on a tumor. So these are two ways by which you can turn your own T cells into better cancer killers. So how does that work then? So look first at that CAR T cell there that has that hybrid molecule now that can recognize something on a cancer cell. For the engineered TCR cell, we gave that T cell a new TCR that it didn't have that can also recognize an antigen on a cancer cell. And if everything works as we hope, then it will kill a cancer cell. Now, what is very important to realize is that a CAR cell can do that multiple times and can live on for days, for weeks, for months, maybe even years. So it is a living drug that can kill many cancer cells. Another way that we can manipulate the immune system to be better at seeing cancer and killing cancer is by working with these antibodies. So here I'm just showing you one out of the many tricks that we can use for that. In this case, we take two antibodies and we turn them into one. So we take the business ends, so the parts of the antibody that can recognize an antigen from both of them, and we paste that together into one hybrid molecule. And if we do that, what we can do then is one part of that hybrid molecule can recognize a T cell, the other part recognizes a cancer cell. And all that that does is bring these cells very close to each other. Turns out that proximity is enough for a T cell to recognize a cancer cell and kill it. And again, these antibodies can be used multiple times, and you can give them, of course, as many times as you want. And that is also what we do when we use this for patients. So these were just a few snapshots of the many strategies that we're working on now. So what is currently already in our clinics, in our pharmacies, basically? CAR cells on this list, you are seeing that there are 5. There are actually 6. We just added a 6th one for myeloma. You can see the ones that are listed here are for leukemia, lymphoma, and myeloma. Trust me that there are many people working on many other cancers also, and also taking it, for instance, to solid tumors. Checkpoint blockade, you see 2 of them listed here, so 2 drugs that could unleash T cells, block the blockers, as I called that. It's mostly used for lymphoma, but those same drugs are again used for many other cancers also. And that hybrid double antibody that I showed you is specifically used for leukemia. And I want to emphasize that this is just a start, right? So we started with bone marrow transplantation, giving us clues that T cells could really kill cancer cells. Now we have found already ways to manipulate T cells to do a better job at that. We're learning about antibodies that we can use for this purpose, but there's much more where this is coming from. There are many more checkpoint blockade molecules. There are many more target antigens that we can target with TCRs. There are many more cells that we could weapon, macrophages, monocytes, NK cells, and so on. So this field, trust me, in the next decade, we're going to see a number of other breakthroughs. And it's not for nothing that in 2013, when it was first clear that this was a true breakthrough, that this was called the biggest breakthrough in all of science. So that is where we are now. I'm sounding hopeful because I really am. I really think that what we're doing at the moment is going to pay off. Thank you very much.
Dr. Vandenbrink, thank you so much for that great news. It sounds like there's a lot of exciting developments happening in blood cancers, leukemia, lymphoma, and even myeloma. We didn't see myeloma on that first slide, but I was glad to see that there is some development there. Now, you mentioned there are now 2 approved CAR T cell therapies for myeloma, one of them brand new, so fast, we didn't even have a chance to update that slide, which I think is really exciting. Just demonstrates how quickly the field is progressing and moving. So on that note of optimism, I'd like to encourage all those who are watching right now, if you have a question for Dr. Vandenbrink, please drop that into the Q&A box on your screen. We'll try to get to as many of those as possible. So we do have a few questions that were submitted while we wait for those to come in during registration. The very first question that we have is, do I need to try chemotherapy before I try immunotherapy?
So the answer is you don't necessarily have to, but most of the use of immune therapy and all of the therapies that we give to cancer patients goes in a certain sequence. And we have to test always if something should be used as a first line, a second line, or a third line therapy. If you look at the current place of most of the checkpoint blockade, CAR cell therapies, and so on, it will be second or third line. So we might see in the next decade that some of these therapies might move into first line. In most cases, first line is still a chemotherapy for most types of cancer.
And can you say a little bit about why that is? If immunotherapy is the greatest breakthrough in cancer treatment, why are we still giving chemotherapy to patients?
Because for many types of diseases, we can actually get a lot of success already with chemotherapy. So if you think about Hodgkin's, for instance, many patients will not need more than several rounds of chemotherapy, and we're actually working to try to make the chemotherapy safer and get more out of less. So that's why we're not ready yet until we have done the phase 3 studies that have shown that certain types of chemotherapy are effective, not necessarily for everybody, that they are less than, let's say, a checkpoint blockade. That needs to be done first with a good phase 3 randomized trial before we move it into first line. This is sort of the natural way that we look at most of the therapies. We start with the late stage diseases and we show there if it's safe and if it has some benefit, and then we see how far forward we can move it in the whole sequence of all of the therapies.
And are we seeing positive results there in earlier stages of disease?
Well, we had an example, for instance, at one of the last ASH meetings when we were looking at the role of CAR T cells for certain types of lymphoma, where the current sequence was that you probably should give these patients first an auto transplant, that actually giving instead immediately a CAR cell therapy seemed to be better than still first giving an auto transplant. So we're seeing these kind of things shifting now.
That's wonderful news. We have a question in from the audience here. It's about different types of lymphoma and whether or not they have different responses. So does immunotherapy work better or worse in different types of lymphoma?
Oh, absolutely. And that is why, if you look at that list again, for different types of lymphoma, we will have to test again. And it might very well be that we have to make, for instance, a different CAR against a different antigen. So there's a lot of work that still needs to be done. CAR cell therapy doesn't cure everybody, and it works in many cases for about half of the patients at the time that we try it. And it doesn't work for every type of lymphoma. So no, we have a lot of work cut out to modify the CAR cells and to test it for all the different types of lymphoma.
Interesting. Another question from one of our audience members who has just received a bone marrow or undergone a bone marrow transplant and wants to know if it's possible to get CAR T cell therapy after that. And if so, is there any increased risk of side effects after receiving CAR following a bone marrow transplant? So several questions in there all at once.
Yeah. So that's a very good question, actually. And initially, when we had patients who had gotten already an allogeneic bone marrow transplantation, for instance, for a form of leukemia, we were nervous about using CAR cells because we were nervous that those CAR cells could cause something called graft versus host. But people have looked now at a number of a series of these patients who got, after an allogeneic bone marrow transplantation, CAR cells made from their own cells, which at that point were donor cells, right? Because they had been transplanted. And we have not seen major risks. We have not seen really a major increase in graft versus host. So CAR cell therapies have a lot of risks also, but so far, we are not excluding a patients from getting CAR cells because they've had a previous allogeneic bone marrow transplantation.
How are those side effects that you mentioned managed? What do they look like, and how are they managed?
Yeah. So the initial story with the CAR cells was that if the CAR cells worked, it seemed, then patients would go through a stage where they were really very, very sick. This was called cytokine release syndrome. Because when people analyzed what was going on with these patients, we saw that there were a lot of cytokines. So there are certain molecules that get into your system during times of activation of T cells and inflammation overall. And patients sometimes had to go to the ICU. And it almost seemed, oh, if that doesn't happen, then the CAR cells won't even work. Well, we know now that that's not true, that there's not a direct link between getting a toxicity and getting efficacy. That's one. The second thing is we have learned much more about picking up any signals of a patient going into that cytokine release syndrome and then start with certain therapies. So we've learned to use a number of drugs now that can specifically block those cytokines from causing that whole syndrome. So we're doing much, much better now in terms of the management of these toxicities.
I see. So doctors are seeing these, they're having a awareness of the possibility of these. As soon as a sign shows up that something bad is happening in the patient, you take care of it.
Yeah, so in this case, often even before patients have any symptoms, we know already from monitoring blood tests that we have to be careful and should start already with one of these therapies, such as anti-IL-6, which is one of the therapies that we use.
Question from one of our audience members is, I have stage 4 lymphoma, non-Hodgkin. What is the longest a patient that has lived who has received CAR T cell therapy that you're aware of?
I'm not sure if I have an exact number for that, but I would not be surprised if we're beyond the 5 years, maybe even further out than that, because the fascinating thing is that with patients who do well initially, right, go into a complete response, there's a fair number of those patients for which that will be durable. So it might very well be that we have patients out there already 10 years without any disease. This is not like for many other cancer therapies that all what you do is a prolonged life. With this kind of therapy, you have actually a chance that some of the people are, I don't like to use the word cured, but that's basically what it is when you're talking about a durable CR.
And CR means?
CR means complete response.
Oh, I see. So almost like a cure, right? Without the U and the E, I suppose. Well, that's good. I think anyone watching, going through this right now would take a CR, a complete response, over anything.
Mm.
What happens if there isn't, since we're talking about responses, what happens when a patient partially responds or doesn't respond at all? Does that mean all CAR T cell therapy no longer is an option for that patient? Is there another type of immunotherapy that patients should look into and talk to their doctor about?
Well, I mean, at that point, of course, the best option is always to be part of a trial. But there are sometimes options to, for instance, give a drug to stimulate the CAR cells to work better. For instance, people are looking at a trick to use those same checkpoint blockade drugs to optimize the activity of CAR cells. So there are all kinds of things that people are working on now, what you could do next to make these CAR cells work better. Sometimes you could even go to another type of a CAR cells. But it's pretty obvious that if that happens, if the initial CAR cell therapy doesn't work, it's always best to do then anything next as part of a clinical trial.
Excellent advice. A question in from one of our viewers here. My daughter is 6 years old and has leukemia. She's eligible for CAR T therapy. Are there long-term side effects I should worry about?
It's a very good question. Of course, a lot of these things we don't know yet, to be honest. At the moment, we haven't seen many of those. So, the types of toxicities that we're typically dealing with, I mentioned already CRS, cytokine release syndrome. In almost all cases, that will pass without any durable consequences. There's a second type of toxicity, which is called neurotoxicity. And that is, of course, the one where we still need to do probably some more research to see if maybe in some cases there is some long lasting consequences from those. But the overall picture is that most of the toxicities that we're dealing with won't last, are not durable.
That's great. Here's another question. I noticed on your sign or your slide, sorry, that there was a lot of leukemia and lymphoma, only one myeloma, but of course now we know there's another. And why is that? Why is myeloma not more easily treated as are leukemia and lymphoma?
Well, first and most of all, myeloma, I would say, is one of the diseases where we had a lot of improved outcomes over the last decade. In my lifetime treating cancer patients, I've seen the life expectancy for myeloma patients double. So there was already a lot of progress in a multitude of other drugs. Some of these drugs actually work also by stimulating immunity. So they're kind of linked to what we're discussing here. Checkpoint blockade therapy was tested for myeloma, and people were very hopeful that that would pan out. Those results have not been so good. So that is why you haven't seen any of that for myeloma. People are still trying to figure out if maybe that needs to be done in a different way. And as I told you already, the checkpoint blockade strategies that we're currently using are only focused on two types of molecules, CTLA4, PD-1. Well, there are many more in the same family of activating molecules and blocking antibodies. So we can definitely hope for some other molecules in those categories to be tested. Finally,
That's
as
fantastic.
we mentioned already, for CAR cells, we have now two types of CAR cells, and there's actually a third one that is not FDA approved, but has also shown some positive data published recently against a different antigen. Those first two were against an antigen called BCMA, but now there's one that is tackling another antigen. So I think we're going to see a lot of progress with different CARs for myeloma. So myeloma is not being left out.
That's wonderful. Of course, yeah. I mean, it's right up there in terms of cancers that all cancers affect us. But I happen to have someone in my family with myeloma, so I like to hear that. Uh, Speaking of another blood cancer on topic, I realize this is a question from the audience. I realize most research is done on the common blood cancers. What hope is there for those of us with rarer forms of disease? I have B cell LPL stage 4. What is B cell LPL, first of all?
So that is also called Waldenström's, and It's a B-cell tumor where you make certain types of antibodies, and specifically if you make a lot of these antibodies, that can lead to consequences. For Waldenström, actually, there's a lot of progress also. There have been some new chemotherapy drugs, but people are also, again, working on making CAR cells for Waldenström's. Fortunately, a lot of these diseases might have common antigens, common targets. So that, for instance, the antigen that was used for many of the CAR cells for lymphoma is an antigen called CD19. That is found on almost all of the B cells that you can find within your blood. So that means that many lymphomas could be treated with the same CAR. So many of these rarer types of lymphoma, for instance, might still have a common antigen that can be targeted with a therapy that was maybe used for a much more prevalent type of lymphoma.
Great. Let's see, another question from a member of our audience. I've been in remission since my stem cell transplant in January 2016. Can I participate in a clinical trial if I'm in remission? I think maybe that's about, is there anything more a patient can do to prevent cancer from coming back? But would be very curious to hear your thoughts on that.
Yeah, it's a very good question. So that is really where it's all about the specifics, right? How long are you out from your transplant? Do your doctors feel that maybe there is a higher risk for you to relapse? Many transplant doctors in such cases might maybe give a patient like that another drug, sometimes drugs, again, to stimulate the immune system, to lower the risk of a relapse. But if this patient is doing fine, is many years out from a transplant, then there's not a direct reason to go into another trial or into another study. There must be some clue, some risk that somebody has to have found or have defined to come up with another therapy.
So if it ain't broke, don't fix it. Is that basically the idea there?
Kind of, yes.
Is it still a good idea, though, for those patients? And maybe this is part of the standard. This is me asking. Maybe this is part of the standard of care that they would go for regular checkups just to make sure everything's okay. And if so, how frequently can someone expect that to happen after they get a stem cell transplant or after they respond well to CAR T cell therapy or checkpoint blockade therapy?
Yeah, so that's all different in every setting. So for bone marrow transplantation, we know that there can be late risks and late complications. So it's always good to have regular follow-up. The frequency of this follow-up is really between you and your doctor, your overall health, and if you had a complication. So I'm not going to put a number on there, but it will be at least every year, maybe more frequently, that you still need follow-up for a number of years after. For the CAR T cell therapy, since it's still relatively new, the FDA wants every patient to be followed for at least 15 years. The background there is that since we engineer T cells, that counts as gene therapy. And of course, people have been very nervous and very careful for many years with gene therapy, because if you manipulate with any gene in any cell, you could cause a cancer. So far, the track record for many years of working with CAR cells and putting genes even prior to that into a T cell is the risk of those T cells causing cancer, turning into cancer, is extremely low. And that's why the FDA lets doctors use this therapy. But they still want to monitor this very carefully, so therefore a 15-year follow-up. For checkpoint blockade, well, many patients will get multiple rounds of a checkpoint blockade, and that's more in the, I would say, the standard follow-up that you would do for whatever cancer they have. It's a little bit less linked to the type of therapy and more to the type of cancer.
So no reason for alarm that your blood cells are going to turn into Franken cells. And okay, no, that's very reassuring. That's good.
Exactly.
Well, we're almost out of time, and we could talk all day about this. It's so fascinating. But before we go, I'd love to know how has treating patients, or how has your perspective on treating patients changed from when you first entered the field to today, and with everything else we're seeing in immunotherapy?
Yeah, I would say it is day and night different. I know that I sounded a little bit like a sales pitch during that talk, but I actually mean that. When I went into this field, it was mostly a palliation. We had a few chemotherapy strategies. We had, of course, a surgery for certain cancers, and some cancers would do well with some form of irradiation, but that was it. And to see this enormous breakthrough that people had worked on for so long, and to see people both with bone marrow transplantation, CAR cells, checkpoint blockade, have durable remissions, something that you rarely see with other therapies, is absolutely amazing. So this is, I feel very fortunate that I can be part of this complete change in cancer care, thanks to these breakthroughs.
Well, that's fantastic, and that makes all of us feel better that so much progress is happening thanks to scientific research, and thanks to doctors like you who are doing this work and helping to save more lives. If you mentioned clinical trials also before as a potential option, so I hope that what those of you who are watching today have learned is to talk to your healthcare team about your options. If you're not already receiving immunotherapy, or if you have been receiving a treatment and it's no longer working, just know from Dr. Vandenbrink that clinical trials might be a great option for you. If you don't know what clinical trials are, or if you want to learn more about how immunotherapy works, be sure to check out yesterday's sessions on the basics of immunotherapy and clinical trials, all available on demand. And with that, thanks everyone for watching. And Dr. Vandenbrink, thank you again so much for sharing that great news with us. We look forward to watching more progress happen in blood cancer.
Thank you very much.